Today, we'll talk about the role, rationale, and application of stem cells in the central nervous system. Stem cells are capable of multiplication, almost indefinite multiplication, and they are capable of differentiation. They are found in almost all tissues. They have two major roles. One is a role in development of organs and tissues, and the second is in repair and replenishment of tissue. Neural stem cells are unipotent cells, differentiate along neural lineages only. Embryologically, they are found lining the neural tube. In the adult, neural stem cells are found in the subependymal layer of the ventricles, in the dentate gyrus of the hippocampus, and lining the central canal of the spinal cord. And then, of course, we have cancer stem cells, which are the growing core of cancers. Let's have a quick look at the embryology of the central nervous system. You have a fertilized zygote. This zygote is totipotent. It divides into placenta and the trophoblast. The trophoblast divides and differentiates into ectoderm, mesoderm, and endoderm. From this ectoderm, in the third week of embryonal life, we have the development of neuroectoderm, which forms the neural plate. This neural plate, the neuroectodermal plate, infolds, creating a neural tube. And this neural tube separates from the ectoderm by a process called disjunction. The neural tube persists into adulthood as the central canal of the spinal cord and as the ventricular system of the brain. There is proliferation, exaggerated proliferation at the cranial end. The lining of the neural tube proliferates. And then these cells, they migrate an inside to out process, whereby the cells migrate to the outside. The next layer of migrating cells go outside, pushing the previous layer in. These neuronal precursors now integrate. How do they integrate? They form connections. They form connections with each other. They form synapses. And they form outgrowths called axons. And these axons also synapse with other neurons. By proliferation of the cranial end of the neural tube, three primary vesicles are formed. These include the prosencephalon or forebrain, the mesencephalon or midbrain, and the rhombencephalon or hind. The prosencephalon consists of the telencephalon, which is the precursor of the cerebral cortex and the basal ganglia, and also the diencephalon, which is the precursor of the thalamus and the hypothalamus. The mesencephalon continues as the midbrain. The rhombencephalon has got two parts. The metencephalon, which forms the pons and the cerebellum, and the myelencephalon, which forms the medulla. How do we use stem cells in neural repair? Endogenous stem cells, as we discussed, are found in stem cell niches. Stimulating factors can be used to stimulate neural stem cells from the stem cell niches. Exogenous stem cells can be used from outside the CNS. These might be embryonal cells. Embryonal cells are usually derived from blastocysts. Multiple blastocysts are discarded in IVF regimes, and those blastocysts can be used. However, there are ethical issues which are precluding the development of embryonic cells. Mesenchymal stem cells are derived from the bone marrow or from adipose tissue. Induced pluripotent cells are now finding favor, and they are achieved by lineage reprogramming of mature cells using retro or lentiviruses. These cells are reinduced into pluripotency and then differentiated into neuronal lineage. What are the routes of administration of stem cells? They can be administered by selective angiography. They can be stereotactically implanted, or they can be administered through the CSF, intraventricular or intrathecal routes. What are the clinical applications of stem cells in neuroscience? In Parkinson's disease, dopaminergic neurons may be transplanted. In spinal cord injury, stem cells may be used, differentiated into oligodendrocytic lineage or into neuronal lineage, where these neurons form bridges across areas of spinal cord transection. In traumatic brain injury, neurons and glial cells may be derived and used. Dementia syndromes also, you can use stem cells along with brain-derived neurotropic factors. In dementia syndromes, we usually achieve nigrostriatal implants, which help in some relief of dementia. Let's have a quick overview of how recovery can occur after a neurological insult. Remember the principles. Acute stroke insults can involve strokes or traumatic brain injury. The mechanism of injury is important. The mechanism includes both inertial injury, where the injury is mostly axonal, and hypoxic injury in a stroke or even when the ICP goes up, where the injury is more cortical. Why? Because the cortex requires a higher concentration of oxygen to sustain itself. Now, is the injury reversible or irreversible? If the injury is reversible by restoration of oxygen supply, then there is a mitochondrial recovery in the neuron or in the cell, which results in a restoration of pumps and functional restoration. This is usually followed by axonal restoration and restoration of synaptic connectivity. In irreversible injury, there are axonal disconnections, and these axonal disconnections may be reconnected either by axonal regrowth or by new synaptogenesis. Some of the delayed recoveries after weeks or months in traumatic brain injury are attributed to reconnections of intact neuronal islands with the core brain by axonal regrowth and by neosynaptogenesis. Neuronal plasticity works when there is cortical loss. Adjacent areas of the cortex take over the function of lost cortical regions. And then we have the stem cells, which can regenerate, migrate, and restore functions by reassimilation. However, stem cell induced recovery occurring naturally is very inadequate for neurological functional recovery. A quick overview: stem cells are capable of multiplication and differentiation. Stem cells may be totipotent, pluripotent, multipotent, or unipotent. Neural stem cells are unipotent. Neural stem cells reside in stem cell niches. These niches are in the subventricular zone, in the dentate gyrus of the hippocampus, in the olfactory groove regions, and around the central canal of the spinal cord. Stem cell therapy may involve stimulation of stem cells in the endogenous niches by various stimulating factors. Exogenous stem cells from outside the CNS may be used. These may be embryonal stem cells, mesenchymal stem cells, or induced pluripotent cells.